Welcome to Lesson 21, Quality by Design. In this lesson, we are going to be looking at the GMPs for the 21st century, the concept of quality by design, quality by design examples, PAT. In 2004, the FDA launched the pharmaceutical GMP initiative called Pharmaceutical GMPs for the 21st Century, a risk-based approach. The link to read this document is in your appendix to this lesson plan. In this document, they explained how they wanted to look at a more proactive approach for management of quality in the pharmaceutical industry. The ICH guidelines, we talked earlier on in another lesson about ICH, the International Conference for Harmonization. At around that time, they issued 3 ICH guidelines. ICH Q8, which is pharmaceutical development, ICH Q9, which is quality risk management, and ICH Q10, which is pharmaceutical quality system. Those 3 guidelines taken together and operated together equals to quality by design, and that is what the FDA were referring to when they talked about GMPs for the 21st century. ICH Q10 then, just to give you an overview of it, it is pharmaceutical quality system. So if we click on our link here, so you can see here the ICH guideline on Q10. So the link to this is also in your appendix. It would be a very good idea to have a look through it and have a read of it so you understand the content. ICH Q10, it describes one comprehensive model for an effective pharmaceutical quality system. So it's based on the ISO quality concepts, the International Standards Organization, and it includes the applicable GMPs as we know them now, and it complements the ICH Q8 and Q9 quality risk management and pharmaceutical development as well. Much of the content of ICH 10 is applicable to manufacturing sites. It's already in our current GMPs. The intent was not to create further expectations, but to consolidate the GMPs. ICH Guideline 8, you can see is pharmaceutical development, and again, the link is in your appendix. It's definitely worth having a read through it again so you have a good understanding of what it is. So quality by design is defined as a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control. It's based on sound science and quality risk management. Quality by design. Part of validation is to demonstrate that there is control of operational conditions, and this is done typically through validation testing. Validation testing establishes process systems is capable of delivering expected results. For quality by design, we're looking at establishing the operational limits of the process so we have greater understanding of how our process works and when something goes wrong with the process, what the impact on our quality attributes will be. It establishes reproducibility as well, so we have good knowledge of our process. It establishes capability and it focuses on preventing inconsistency. What it is not, quality design is not just a means of putting in more finished product testing. The idea is we have a better understanding of our process and we're making more scientific judgments on how we're managing our process rather than just doing more testing. So it's not quality by testing, it's quality by design. And just to emphasize that point again, we are designing our process to ensure quality. We're not testing our product to ensure quality. There's an interesting document here describing the differences between process validation and design space verification. Design space verification should not be confused with process validation, even though both take into consideration prior knowledge and are conducted on a commercial scale. However, the scope of both is not the same. Process validation demonstrates consistency of the process at normal operating ranges. Design space verification demonstrates that the scale effect and/or the model assumptions are under control in the new area of design space and do not affect the product quality. Unlike validation, which covers all the steps of the manufacturing process, verification studies refer only to those operations where a design space has been proposed. Just to note, continuous process verification is an alternative approach to traditional process validation, in which manufacturing process performance is continuously monitored and evaluated, and that's from the ICH Q8. So quality by test, and that's a fixed process. We put in our materials, we have our process parameters, and our product comes out as a variable product. So we have our normal Gaussian curve. Some stuff will be in spec, some may be out of spec, depending on how well your process is running, but you're not altering any of your process parameters. Everything is fixed. In quality by design, it's a more controlled process. You're controlling the materials that you're putting in. You've got set attributes for them that you will accept. You are looking at your process parameters. If your process is going towards your operating ranges, the edges of your operating ranges, you're making adjustments here, and that's monitored and controlled all of the time. And often you would be using process analytical technology to do that. Your product then, when it comes out, is your green line here. So you've got a much sharper Gaussian curve, and it's much more controlled. It's much more within specification, so you shouldn't be seeing outliers or product that's defect. What to control and what to impact. So we would be impacting, we've got our critical to quality, we've got our critical materials attributes, our CMAs, our CPPs, which we talked about before, your critical process parameters, your CQAs, your critical quality attributes. So what are we looking at here? We're talking about our critical material attributes here. We need to control those, and we've specified what they need to be. If we see an alteration in our material attributes, we're going to know in advance what the impact is. If the particle size is slightly larger or slightly smaller than normal, it might still be in specification, but it might mean we might need to make alterations to our process parameters to allow for this. And again, that would be all contained within our design space. The other thing we need to look at controlling for our inputs are our critical process parameters. So based on our materials and our process parameters, we should be getting our output, our critical quality attributes should remain constant then as a result of managing our process and our materials going in. So our CPPs, critical process parameters. Critical process parameters, they are the process parameters whose variability impacts the product quality. And we talked about those before, so you'll be familiar with the term. They must be controlled and monitored to ensure that the process produces the desired quality. If we don't have control over our CPPs, we're not going to have a control over our critical quality attributes. CPPs have a direct impact on our CQAs. So not managing our CPPs means that our critical quality attributes will have a high variability. Examples of critical process parameters for small molecules. So small molecules are our API molecules. So they would be things like temperature, addition rate, the cooling rate, rotation speed, physical parameters like that. Examples of critical process parameters for large molecules, so that's the biopharma, would be things like temperature, pH, your agitation, your media, your feed type and rate, and your sterility. The critical quality attributes are the properties of the product that can characterize its quality. A CQA is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit and distribution range. It must be monitored and controlled to ensure the desired product quality. The CQAs for small molecules, again your API, typical CQAs for an API might be things like your particle size, your impurities, your release profile, your specific volume, your moisture level, again sterility, content uniformity could all be critical quality attributes depending on your product for an API. Critical quality attributes then for a large molecule in the biopharmaceuticals could be things like your composition, so your structure, your protein content and your purity, etc., the concentration of it, the aggregation, the glycosylation, the pH, the appearance, the impurities. The impurities could be advantageous, so you've got viral purity, microbiological purity, bioburden in your mycoplasma. You could also have things you don't want like endotoxins or leachables that come from the process unit operations. So when we're talking about design space, the things that we can make variable are our materials, making sure that we've got our critical material specifications going in, and our process parameters, our CPPs, so we can manage those within our design space. Obviously, this would need to be filed in advance as part of our regulatory filing. You can't decide to do design space once something has already been filed unless you make alterations to it via a change control process. The overall concept and steps. So the first thing we need to do is define, well, what is our desired product quality? And once we know what that is, it'll be a lot easier for us then to identify the CQAs, the critical quality attributes. When we've got our CQAs, we identify the critical process parameters that affect our CQAs using a risk assessment methodology. So we're going to be looking at that in a further on lesson. We execute some experiments and construct our design space using statistical analysis and methodology. So it's very science-based and it's not intuitive, it's not guesswork, it's all based on facts and figures and science. We implement controls to ensure consistency of the product quality and quality risk management as well. Our knowledge space is this big circle here. So this is what we know about. If we affect critical process parameter A, then we know that the impact is going to be B on our critical quality attribute. If we have change to our material attribute going into our process, then the effect is going to be C, and so on and so on. So we have a good knowledge of that whole space. What happens inside here? If we make a change to one thing, what is the impact on everything else? And what can we control? Within the knowledge space, we've got our design space, and within our design space, we have our normal operating range where our process normally is working. Working within the design space is not considered as a change. The design space is proposed by the applicant and it's subject to regulatory assessment and approval. Establishment of design space is to understand the product and the process better. When we present our design space, you're going to end up with something graphical that looks something like this. So if you have a read of the ICH Q8, it gives some really good tangible examples of how that works in practice. So just to give you an overview of them here. So this example, your design space is determined from the common region of successful operating ranges for multiple CQAs. So you're basically overlaying your graphical plots to show where your design space is and where your safe space is for working. So in this example, you've got two CQAs, two quality attributes, and those are your tablet friability. So that's how easy your tablet will chip when you put pressure onto it, and your dissolution. And we've got two process parameters from a granulation operation that affect these two CQAs. So our two CPPs, our critical process parameters, are 1 and 2, and our two CQAs are the tablet friability and the dissolution. Figure number 2 here, this shows us our dissolution is greater than 80, so that's what we want. We want a dissolution of greater than 80%. So our white space here is where we want to be working for our dissolution versus parameter 1 and parameter 2 when they're plotted on a curve such as this. Figure 2b, we've plotted our friability. So our friability needs to be less than 2%. So again, we've got our white space here. This is the friability that we want. This is our approved acceptable result for friability when you plot it against parameter 1 and parameter 2. Parameters 1 and 2, they're factors of the granulation operation that affect the dissolution rate of the tablet. So it could be something like your excipient attribute, your water amount, or your granulation size. So they're just examples. So the design space is determined from the common region of successful operating ranges for multiple CQAs. They've taken the two critical quality attribute plots and they've laid them on top of each other. So you've got parameter 1 here and parameter 2 here. And our white spaces, when they're laid on top of each other, this was our friability, if you remember. It was this section, and our dissolution was this curve here. So when we put the two together, this is the space that we can operate within. This is our design space. So figure 2c shows the overlap of these regions and the maximum ranges of the proposed design space. So as an applicant, you can elect to use the entire region that you see here in the design space, or you can just use a subset of it. This is a second example. So this is again from the ICH. So it's a design space for a drying operation. So the process is dependent on the path of temperature and our pressure over time. The endpoint for the moisture content is 1 to 2%. So you'll see our endpoint is here in the green line. Our time is plotted along this, and our moisture content is plotted along this line. So if we operate above the upper limit, so our upper limit is here, that will give us excessive impurity formation. And if we operate below the lower limit, that can give us excessive particle attrition. So we have to operate within our design space along here. That tells us where we can operate. Process analytical technology. In the past, we would have used all offline analysis, which meant taking a sample from your batch, sending it to the labs, and maybe waiting for a couple of days to get the results back. At that stage, your batch was probably finished, and if there was a problem, the only thing you could do is potentially rework it if that was feasible, or else end up having to segregate it or reject a batch if it wasn't up to quality. PAT is process analytical technology where you've got your testing in situ in the process, so you're getting results instantaneously. The type of testing you can do with PAT is particle size analysis, high performance liquid chromatography or HPLC, your atomic absorption or infrared spectroscopy. So all of those give you automatic, immediate results from your batch, so you can take more immediate action and potentially you will be able to make alterations to your critical process parameters to prevent any issues with your critical quality attributes. So in theory, you should be managing your process an awful lot better. You should be ending up with a lot less rejects or defects. So the advantages of it is that you've got real-time analysis, so you've got effective in-process quality control, and it's more cost-effective as well. It's also less prone to process variation. So over to you. So what is QBD? How is it different to process validation? What is the design space and how would you ascertain it? How can PAT be used in a process to support QBD? Thank you very much for your time and attention. We'll see you for the next lesson.